Good day, this is Professor Will, CBMD PhD. Today's Thursday, June 25th, 2020, and it is 1:46:24 a.m. Eastern Standard Time, and this is a continuation of my corona research. But before we get started, let's take a look at my message of the day, and a little something I recently wrote. Today, presenting my video diary, I would like to mention that just a couple days ago, during an outdoor walk, I discovered a small yellow leaf lying on the grass. It was covered with red lumps which looked to me like some sort of environmental damage. I immediately picked up the leaf for examination, which I call Operation Specimen Number 1. First, I put it under my large magnifying glass and took a stereoscopic photo using a mobile device. Then later on, I put it under my compound microscope for thorough investigation. During my assessment, I wiki-paged Specimen Number 1, which described my find. They are called drwillphd, as a family of more than 200 genera of mites, which live as plant parasites commonly causing galls or other damage to plant tissues, and hence known as gall mites. As the months progress throughout summertime, I'm going to slowly investigate these fields of natural exposures, and especially the nature of parasites pertaining to our environment. Well, I hope you enjoyed listening, so let me continue on with my research.
Far UVC light safely kills airborne coronaviruses, study finds. Date June 24, 2020. Source Columbia University Irving Medical Center. Summary. A type of ultraviolet light called UVC, which is safe to use around people, kills more than 99.9% of airborne coronaviruses, a new study has found. More than 99.9% of seasonal coronaviruses present in airborne droplets were killed when exposed to a particular wavelength of ultraviolet light that is safe to use around humans. A new study at Columbia University Irving Medical Center has found. Based on our results, continuous airborne disinfection with far UVC light at the current regulatory limit could greatly reduce the level of airborne virus in indoor environments occupied by people. Conventional germicidal UVC light, 254 nanometer wavelength, could be used to disinfect unoccupied spaces such as empty hospital rooms or empty subway cars. But direct exposure to these conventional UV lamps is not possible in occupied public spaces, as this could be a health hazard. To continuously and safely disinfect occupied indoor areas, researchers at Columbia University Irving Medical Center have been investigating far UVC light, 222 nanometer wavelength. Far UVC light cannot penetrate the tear layer of the eye, or the outer dead cell layer of skin, so it cannot reach or damage living cells in the body. The researchers had previously shown that far UVC light can safely kill airborne influenza virus. A new paper extends their research to seasonal coronavirus, which are structurally similar to the SARS-CoV-2 virus that causes COVID-19. In the study, the researchers used a misting device to aerosolize two common coronaviruses. The aerosols containing coronavirus were then flowed through the air in front of the far UVC lamp. After exposure to far UVC light, the researchers tested to see how many of the viruses were still alive. The researchers found that more than 99.9% of exposed virus had been killed by a very low exposure of far UVC light. Based on their results, the researchers estimate that continuous exposure to far UVC light at the current regulatory limit would kill 90% of airborne viruses in about 8 minutes, 95% in about 11 minutes, 99% in about 16 minutes, and 99.9% in about 25 minutes. Using far UVC light in occupied indoor spaces, the sensitivity of the coronaviruses to far UVC light suggests that it may be feasible and safe to use overhead far UVC lamps in occupied indoor public places to markedly reduce the risk of person-to-person transmission of coronaviruses, as well as other viruses such as influenza. Ongoing studies in SARS-CoV-2. In a separate ongoing study, the researchers are testing the efficiency of the far UVC light against airborne SARS-CoV-2. Preliminary data suggests that far UVC light is just as effective at killing SARS-CoV-2. Far UVC light doesn't really discriminate between coronavirus types, so we expected that it would kill SARS-CoV-2 in just the same way. Since SARS-CoV-2 is largely spread in via droplets and aerosols that are coughed and sneezed into the air, it is important to have a tool that can safely inactivate the virus while it is in the air, particularly while people are around. Because it is safe to use in occupied spaces like hospitals, buses, planes, trains, train stations, schools, restaurants, offices, theaters, gyms, anywhere that people gather indoors. Far UVC light could be used in combination with other measures, like wearing face masks and washing hands, to limit the transmission of SARS-CoV-2 and other viruses. Tuberculosis cases and deaths predicted to spike due to COVID-19. Date June 24, 2020. Source: London School of Hygiene and Tropical Medicine. Summary. A new study estimates that at least 110,000 additional deaths from tuberculosis in China, India, and South Africa unless health services maintain and strengthen. The global COVID-19 pandemic can significantly increase the global burden of tuberculosis (TB) due to disruptions to health services and delays to diagnosis and treatment, according to new estimates published in the European Respiratory Journal. Before COVID-19, over 4,000 people were dying from TB every day. In countries where health services are strained, there is a fear that TB cases and deaths could rise significantly. Researchers from London School of Hygiene and Tropical Medicine, LSHTM, and Lancaster University estimate additional TB deaths and cases in China, India, and South Africa over the next 5 years. They examine the impact of various reductions in social contacts and impacts on health services due to COVID-19. The researchers hypothesize that social distancing might reduce TB incidence as Mycobacterium tuberculosis, the bacteria which causes the disease, is transmitted via droplets in the air, similar to the coronavirus. However, even after taking into account this potential reduced TB transmission, the most likely scenario is estimated to result in more than 110,000 additional TB deaths. In the worst case scenario, where the impact of COVID-19 on health services is severe, this number could rise up to 200,000 additional deaths. There is concern that the COVID-19 pandemic is resulting in decreased TB clinic attendance, delayed diagnosis and treatment. This is especially so in low and middle income countries where health services or access to them might be sustainably disrupted. Early anecdotal information from China, India, and South Africa suggests that the number of people being diagnosed and treated for TB has fallen significantly. The team measured the effect of these factors on TB incidence and deaths in China, India, and South Africa, 3 countries which make up approximately 40% of global TB cases. They modeled different scenarios with various implementations of social distancing measures and health services disruptions lasting for 6 months to estimate the impact of COVID-19 on global TB burden. They found that if there is substantial health service disruption, such as 50% reduction in TB cases detected and successfully treated, there would be an increase in both incidence and deaths from TB over the subsequent 5 years, regardless of the level of social contact. The most probable scenario, based on the current information and distancing measures, suggests 6,000 additional deaths could occur in China, 95,000 in India, and 13,000 in South Africa. However, if social distancing measures are minimal and there is substantial healthcare disruption based on 80% reduction in case detection and treatment success, the model predicts an 8 to 14 increase in cumulative deaths from TB over the next 5 years. We need to act now to ensure innovative approaches to people-centric TB care are the focus, so that the fight to end this pandemic doesn't overwrite the hard-won gains made against this disease. Given that health service disruption far outweighs the benefit of social distancing, it is crucial to maintain and strengthen TB-related health services during and after COVID-19. It is also important to note that the decline of health services is likely to have a greater impact on drug-resistant TB patients, as they often require longer treatment. Additionally, we examined social distancing measures over a 6-month period, but with the potential of subsequent waves of the virus, the impact could last longer. The authors acknowledge limitations of their work, including that it did not take into account setting-specific differences within the countries, while the effect on service delivery needs to be investigated in greater detail. It also did not incorporate external factors of the COVID-19 pandemic, including increased poverty or reduced access to antiretroviral treatments in the areas of high HIV prevalence. Further research is required to examine these factors, as well as whether TB patients are at higher risk of severe COVID-19. Bacterial predator can help reduce COVID-19 deaths. Date June 24, 2020. Source: University of Birmingham. Summary. A type of virus that preys on bacteria could be harnessed to combat bacterial infections in patients whose immune systems have been weakened by the SARS-CoV-2 virus that causes a COVID-19 disease, according to new research. Called bacteriophages, these viruses are harmless to humans and can be used to target and eliminate specific bacteria. They are of interest to scientists as a potential alternative to antibiotic treatment. 2 strategies are proposed where bacteriophages could be used to treat bacterial infections in some patients with COVID-19. In the first approach, bacteriophages would be used to target secondary bacterial infections in patients' respiratory systems. These secondary infections are a possible cause of the high mortality rate, particularly among elderly patients. The aim is to use bacteriophages to reduce the number of bacteria and limit their spread, giving the patient's immune systems more time to produce antibodies against SARS-CoV-2. By introducing bacteriophages, it may be possible to buy precious time for the patient's immune systems, and it also offers a different or complementary strategy to the standard antibiotic therapy. In the same way that we are used to the concept of friendly bacteria, we can harness friendly viruses or phages to help us target and kill secondary bacterial infections caused by a weakened immune system following viral attack from viruses such as COVID-19. This is not only a different strategy to the standard antibiotic therapies, but more importantly, it is exciting news relating to the problem of bacterial resistance itself. In the second treatment strategy, the researchers suggest that synthetically altered bacteriophages could be used to manufacture antibodies against the SARS-CoV-2 virus, which could then be administered to patients via a nasal or oral spray. These bacteriophage-generated antibodies could be produced rapidly and inexpensively using existing technology. If this strategy works, it will hopefully buy time to enable patients to produce their own specific antibodies against the SARS-CoV-2 virus, and thus reduce the damage caused by an excessive immunological reaction. The research focuses on the identification and development of bacteriophages that kill pathogens in an effort to develop new antimicrobial. We can also exploit our knowledge of phages to engineer them to generate novel and expensive antibodies to target COVID-19. This clearly written article covers both aspects of phage biology and outlines how we might use these friendly viruses for good purpose. This pandemic has shown us the power viruses have to cause harm. However, by using beneficial viruses as an indirect weapon against the SARS-CoV-2 virus and other pathogens, we can harness that power for a positive purpose and use it to save lives. The beauty of nature is that while it can kill us, it can also come to our rescue. It is clear that no single intervention will eliminate COVID-19. In order to make progress, we need to approach the problem from as many different angles and disciplines as possible.
Good day, this is Professor Will, CBMD PhD on infectious disease, molecular microbiology, and pathophysiology. I hope you enjoyed listening. Until next time, have a great day.